Good evening. I just want to welcome you all to the 995th meeting of the Boston Society. Tonight's lecture will be on neurology of consciousness, which I think will be pretty darn exciting. We'll, I'm sure, stay awake for that. I just actually say this is a first. I had someone ask me if we could video this so they could see it in Europe. So I don't know if that's happened before, but it's a first. Yes. So I just wanted to remind you that we have some good programs coming up. The next one is going to be about neurology of empathy, and I think it should be really fun. And that's going to be, Bruce, is that going to be over at your... At the Mass General. The other ones are going to be all here, but we have some really good speakers. We're going to end our 1000th anniversary, just happens to be on the Denny Brown night, and it's going to be a movie of, it's an old movie of Denny Brown doing a brain cutting. I hear it is a classic. He's smoking and cutting, and we're going to have, it's going to be like a movie, popcorn, movie, kind of venue. So that's all I have to say. I think Dr. Galberto was nice enough to introduce Dr. Saper.
So everybody who has been here for the past 20 years must have heard about Cliff Saper. He's the chairman of the Department of Neurology at Beth Israel Deaconess Medical Center. He trained in Wash U and then Cornell, and then was working both at Wash U and the University of Chicago before he came here 20 years ago, which time the neurology departments were very different. They were small, undifferentiated, without fellowships, just residencies. And he changed all that and made it into a modern department. He recruited some young Turks who have now grown to become full professors and have their own research and clinical programs, and we remain one of the strongest departments around the country thanks to the energy of Cliff's. He, of course, is very well known for his research on the hypothalamus and the brainstem control of autonomic functions, including sleep, appetite regulation, temperature, and things like that. He's been editor of the Journal of Comparative Neurology until very recently, and he will be the next editor of the Annals of Neurology. And so it's with great pride and honor to introduce Cliff.
Thank you, Al. It's wonderful to be introduced by. I've known Al for many years and was aware of his work long before I came here, and so it's wonderful to have friends like that to introduce you. I also always like to talk in this room because of the portraits on the wall. So many of you are probably not familiar with this room, and I just would point out my two favorite portraits on the wall. It's not Jean Brownwald over there, by the way. Up in the corner is Harvey Cushing, and Harvey is looking at a skull X-ray, and if you look closely, you'll see that the patient has a blown-out pituitary fossa. Yeah, that's a terrific painting. The other one I really like is on the wall over there. It's the fellow who's sort of bald in a white coat with a stethoscope in his pocket, standing at the bedside. That's Soma Weiss. And Soma Weiss was a chair of medicine, I believe, at the Brigham right before World War II. And he was a brilliant young physician in those days, not a neurologist, but he studied neurological phenomena, particularly carotid sinus reflexes, and described carotid sinus syncope in really classic papers. And then he was at City Hospital and he did that. Then he came to the Brigham. Was recruited here at a very young age, something like 40 when he became the chief of medicine here, and sadly died of intracerebral hemorrhage a few years later. But I always like his portrait as well. So I thought I'd point out some of the people that make this a nice room to have for the Boston Society. I also want to, with Nico today, honor one of our own mentors. We're going to reprise a duet that we gave a few weeks ago in New York. And this is a hopefully this will work. Here we go. So this was at a symposium in the memory of Fred Plum. And Fred plays an important role in both of our work, which we'll tell you about. He was the chief of neurology at Cornell when both of us were residents there. I was a resident there from 78 to 81, and then Nico from 93 to 96. So two different eras there, but Fred was there the whole time. He was chief from about 68 to about 97 or something like that, almost 30 years. And he was one of the great icons of the field as well. And he, early in his career, wrote a book that changed the way medicine is practiced in the course of my career. And so that's the book. This is actually the third edition, but the first edition of The Diagnosis of Stupor and Coma, great title for a book, certainly a page turner, came out in the late 60s. And when I was in medical school, we had to read this book at Washington University. We had to read it twice. Once when we were in our second year pathophysiology course, and then again when we were in our clinical year. And I can tell you that this book begins with a chapter on the neurobiology of consciousness and coma. And I did not understand it at all when I was a second-year medical student. By the time I was a third-year medical student, I was sort of beginning to think about it. Then when I was a resident at Cornell, they were writing the third edition of this book, the one that's shown there. And so Fred, who knew that I had some interest in this field and had a background in neuroanatomy, asked me to review the basic science in that chapter on the neurobiology of consciousness and coma. And I became convinced at the time that much of what we had been taught in school was probably not correct, and that the evidence for a lot of the important pieces of this puzzle were missing. And so I helped write that first chapter, and as a result, I got a copy of the book from Fred and Jerry Posner with this inscription on it, which I hope you can read. It says, Cliff, many thanks for your help, Jerry and Fred. So that book is one of my prized possessions. And many years later, after I'd been working on this problem for quite a long time, I had the pleasure of working with them again on the last edition of the book, which I'll describe a little bit later. But back in 1980, when I was a resident, I had the opportunity to start working on understanding the neurobiological basis of consciousness. And this was generously provided by Dr. Plum, who I came to him with a totally harebrained idea. I said, I think that most of what is in this chapter in this book is wrong, and I'd like to figure out what's right. That we really needed to restudy the ascending arousal system. And would you let me do this as a third-year PGY4 resident? Would you let me have half a year to do that? And he actually said yes. And as a result of that, I was able to get enough preliminary data to get my K08 award and set my career off in that direction. So what did we find when we did these experiments? So I'm going to summarize a lot of data for you in a relatively few summary slides, and then I'm going to tell you why much of this is probably not right. So this is sort of the traditional, well, this is the current synthesis of how the arousal system works. Those of you who were trained back in the era that I was would have been told about a mysterious entity called the reticular activating system. There were supposed to be neurons in the reticular formation that activated the thalamus and caused wakefulness to occur. And it turns out that none of the above is correct. So it turns out that the arousal systems, at least the ones that we knew about as of a couple years ago, are not in the reticular formation at all, but are actually in well-defined cell groups that are shown here. And there's basically two classes of them. One class dominated by cholinergic neurons in the pedunculopontine and laterodorsotegmental nuclei at the junction of the midbrain and pons, send their axons to the thalamus, where they activate the thalamic relay nuclei, inhibit the thalamic reticular nucleus, which blocks thalamocortical transmission, and basically open up the gate so that information from the sensory systems relayed through the thalamus can get to the cerebral cortex. This system is sometimes equated with the arousal system, but it actually is not capable of causing wakefulness by itself. Rather, it requires the concerted activity of this system in red. And the red system begins with neurons that are monoaminergic, locus coeruleus noradrenergic neurons, raphe serotonergic neurons, some dopaminergic neurons in the ventral periaqueductal gray, and histamine neurons in the posterior part of the hypothalamus, in the tuberomammillary nucleus. And all of these monoamine systems have the property that they fire most during wakefulness. They slow down as you go into slow wave sleep, and they stop firing completely during REM sleep. All of them also project directly to the cerebral cortex, bypassing the thalamus. They're joined by neurons in the hypothalamus that contain the peptide orexin, which is found in the lateral hypothalamus, and by basal forebrain neurons that contain either acetylcholine or GABA. Orexin is an arousal producing neurotransmitter, as I'll show you in a minute. The basal forebrain cholinergic neurons innervate cortical pyramidal cells and turn them on. The GABAergic neurons innervate cortical GABAergic interneurons and disinhibit the cortex. So all of these red pathways together, which are interconnected with one another, result in activating the cortex and making it ready to receive information from the thalamus. So when all of those pathways are firing simultaneously, you're awake. And as you go into sleep, they all slow down together. The cholinergic system stops. And then finally, intermittently, when you go into REM sleep, the monoamine systems stop completely, but the cholinergic system turns back on. It transmits thalamic information to the cortex, but the cortex is woefully unprepared for it. And the result is that you have a dreamlike state where the cortex is receiving information, is activated, but can't process it efficiently, and isn't able to produce consciousness. Consciousness really requires both of these systems. At least that's the going theory. I'll come back to that in a minute. Now, our group was also interested in how this system gets turned off when you go to sleep. And so in the mid-90s, shortly after I got to Harvard, one of my graduate students began studying this, and he ran across the pathway that's shown here in purple. It begins in the preoptic area, the hypothalamus, the ventrolateral preoptic nucleus. And these are inhibitory neurons. They contain GABA as well as an inhibitory peptide, galenin. They send their axons to all the components of the arousal system, and they turn the arousal system off in a coordinated way. And basically, there are very few sites that have this capability that project to so many of these systems directly. And this seems to be a key site for turning the brain off and allowing sleep to occur. If you were to lesion those neurons in the ventrolateral preoptic area, animals sleep about 30 to 40% less. So for a human, that would be 2 or 3 hours a night of sleep less because those neurons were gone. They're not the only neurons involved in sleep control, but they play a critical role, and no other cell group has been found that plays as important a role in turning off the arousal systems during sleep. Now, these sleep-producing neurons turn off the arousal systems during sleep, but it turns out they also receive inputs from the arousal systems, and these inputs, particularly from the monoamine cell groups, inhibit the ventrolateral preoptic neurons. So essentially, both sides of the system are inhibiting each other, and this produces the conditions for what electrical engineers would call a flip-flop switch. Are there any electrical engineers? There's a couple of you. Matt, you're not an electrical engineer? Okay, well, anyway, this was pointed out to me by one of my graduate students who was an MIT graduate in electrical engineering. And he thought this was so elementary that everybody should simply know this because you learned this in your first course in electrical engineering. But if you want to design a circuit that makes rapid transitions between two states and doesn't hang around in the middle, you design a mutual inhibition where each side turns off the other. So if you could imagine what's going on in the brain during a normal day as you build up increased sleepiness by being awake for a long time, as your circadian propensity for sleep changes as you get to the night, you might drift back and forth between an awake and a sleep state if you were on a seesaw like is shown here. But instead, what happens is that people are awake for most of the day, and then they go to bed, and in a few minutes they're usually asleep. And then when the clock goes off or when they decide to wake up in the morning, they wake up. And the transitions are usually quite rapid. Now, that doesn't mean they're instantaneous like an electric switch, but in general, the transitions in humans take a few minutes. And these are less than 2% of the total day. You spend 98% of your day either awake or asleep, and only 1 to 2% in the transition between the two. And that's a very important component of being able to respond to things in the environment. You wouldn't want to be walking around all day in an intermediate state, half asleep, and you'd be run over by a bus on Longwood Avenue. So this is actually a mechanism used by the brain. It turns out to be a motif used in many circuits in the nervous system where you need to flip from one state to the other relatively rapidly. Now, one of the problems with that kind of a circuit is that it might be a little twitchy, so it might flip at the wrong time. And you wouldn't want, for example, if you were driving down a road at night and it was dark out and monotonous, to fall asleep at the wheel because you had suddenly flipped from an awake to a sleeping state. Or when there was a sound in your house at night, you wouldn't want to flip into a wake state and be trapped there and be unable to get back. And so the brain has developed an additional system that consolidates sleep and wakefulness. It makes the switch less twitchy. And the way it does this is by using neurons that contain the neurotransmitter orexin, sometimes called hypocretin. And these are neurons in the hypothalamus, in the lateral hypothalamus, that activate the arousal systems. And in doing so, they consolidate wakefulness, resulting in, during the night when you're asleep, they're turned off, the arousal systems are turned off, and you get consolidated sleep. And in patients who lack these neurons, who have the disorder we call narcolepsy, they lack the ability to maintain wakefulness in a daytime, which everybody is aware of, I suspect, in this room. However, what most people aren't aware of is that they also lack the ability to maintain sleep. They tend to wake up too much at night when they're asleep. And it's hard to imagine why a wake-producing neurotransmitter loss of that transmitter would make you wake up too much at night. But in the flip-flop model, it actually is predictable mathematically from the way the model works, that if you lose this extra strength on the on side of the switch, that both sides of the switch will become weaker and more unstable. So you're going to have more transitions in both states. So this is the current model for the ascending arousal system and the sleep system. And this is where I came to it and began wondering, of all of these cell groups, which ones are really critical for maintaining wakefulness? And the problem with this model is that you can lesion any one of these arousal-producing cell groups, and the amount of wake sleep is pretty much the same. So many people are surprised. If you remove the locus coeruleus, we've done this in the lab, you couldn't tell the difference. Animals have exactly the same amount of wake and sleep. They have no extra transitions. They just behave like totally normal animals. It doesn't seem to matter to them at all. You can take out the histamine neurons, the serotonin neurons, even the orexin neurons. You're a little sleepy during the day, but you don't have more sleep. You just have a little instability, so you have some of your sleep during the day and some of your wake at night. But you don't actually have more sleep. You don't lose wakefulness. So it wasn't at all clear which of these arousal systems were really important. Also, there's this gold pathway and this red pathway. And it's been thought for many years that the thalamus is really what's important in causing the content of consciousness, and that should be reflected in the EEG. The EEG should be is the electrical activity in the cortex, after all. The thalamus has massive cortical connections. thalamic neurons undergo oscillations that look a lot like the kind of waves that occur in the cortex. They oscillate, they're firing patterns. And so many people think that the thalamus causes the normal waking EEG pattern. And so we began to wonder if this was the case. So we decided to see what would happen if we removed the neurons in the thalamus. And we did that by use of a neurotoxin that kills nerve cells. And this is an example of one of those experiments. These were done in rats, by the way. I'll tell you in a minute our observations in a human case. So in this rat, you can see the thalamus is essentially cored out of the brain. There's a few neurons left in the lateral and medial geniculate back here, but almost everything else is gone. And this rat walks around its cage, or it did when it was alive, and you would not tell the difference between this rat and a normal rat. They just simply look normal. They have normal wake-sleep cycles. If you look at the cortex, we stain the brain for a protein called CFOS, which is expressed in neurons when they're active. And the cortical neurons have an identical amount of CFOS protein. The arousal systems have an identical amount of CFOS protein. And the EEG is indistinguishable from a normal animal. Indistinguishable. Now, I'm not trying to say the thalamus doesn't do anything. So let me tell you about a human case that we saw who had bilateral thalamic hemorrhages. The patient with bilateral thalamic hemorrhages lacked the content of consciousness. And Nico will tell you more about the content of consciousness in his talk. So she was in a persistent vegetative state. She went through wake-sleep cycles, but when her eyes were open, she did not respond to anything in the room. Maybe occasionally a rapid movement would cause a saccade, which is a brainstem-mediated saccade, but nothing that you could look at that was cortical function. So the cortex does require the thalamus in humans, the content does, but amazingly her EEG was also very close to a normal patient, a little encephalopathic. Her waking EEG was in the 6 to 8 hertz range predominantly. Her sleeping EEG during slow wave sleep was in the 1 to 3 hertz range. And she went through cycles like any normal person without a thalamus at all. So a lot of what we think of in the EEG is reflecting what's going on in the cortex, is reflecting the arousal system in the brain. But that arousal system really doesn't involve the thalamus very much. And the thalamus, to the extent that it contributes the content of consciousness, is extremely important. But it actually doesn't play a huge role in determining the level of consciousness, a big difference. So we wondered what might actually provide that. And so we decided to go for the red pathway, and we decided to go for the basal forebrain, which is its terminus. And we did that because no one had really tried wiping that out yet. People had done the orexin neurons, the histamine neurons, the locus coeruleus, and none of those really had much effect. So we went for the last node in this chain. And we placed lesions there, and I'll tell you about that for a minute. So one set of animals, we got rid of the cholinergic neurons in the basal forebrain, and we did that by using a selective cholinergic toxin. So the way this works is that it has an antibody against the nerve growth factor receptor, which is found in the basal forebrain cholinergic neurons. This is the IgG192. It's a monoclonal antibody, and it's linked to a toxin called saporin that targets it just to those cells. And when you inject it into the brain, I don't know how well you can see this in back, but there's supposed to be a lot of brown neurons in the basal forebrain. And in this animal, there's 2 left. And all of the other neurons are still there. This doesn't kill all the cells, it just kills the cholinergic cells. So you wipe out the cholinergic system in the basal forebrain, and the animal looks completely normal. Its cortex shows the normal amount of FOS during wakefulness. The locus coeruleus and the histamine neurons arousal system shows FOS during wakefulness. The animals essentially are normal in that experiment. You go on. So then we decided to use a higher dose of this toxin. At a high dose, the toxin begins to be taken up by, we used a high dose of a toxin called orexin soporin. I'm sorry, let me go back a slide. I apologize, I got these slides out of order. So this is a slide showing the use of a low dose of orexin soporin. This is a toxin that spares the cholinergic cells and kills everything else. I apologize for getting this out of order. So this is the basal forebrain stained with a brown antibody against choline acetyltransferase. The cholinergic neurons are there, but none of the other neurons are present. It's just cholinergic neurons left. And again, these animals, when you take out all the other cells, the cholinergic cells support normal cortical wakefulness, wakefulness throughout the system. The animals look completely normal. If you use a high dose of orexin soporin, you kill all the cells in the basal forebrain. So that's this experiment. And in this experiment, we killed all of the cells, both cholinergic and non-cholinergic, in the diagonal band, in the medial septum, in the basal forebrain, substantia nominata. And this is a photograph, and there's like 1 or 2 cholinergic cells left, but everybody else is dead. And interestingly, when you kill all the basal forebrain cholinergic cells and all of the other basal forebrain cortically projecting neurons, the animals actually fall into a deep coma. So without the basal forebrain node, there's no cortical FOS expression. The EEG shows a high voltage slow wave. If you look at a power spectrum, it's all in the 1 to 3 hertz range. It's monotonous delta EEG. Interestingly, the arousal systems are trying to turn the brain on. The locus coeruleus, the histamine neurons are active. They cannot keep the brain awake in the absence of the basal forebrain. So the basal forebrain, combination of cholinergic and non-cholinergic neurons, seem to be necessary for maintaining a waking state. So these basal forebrain lesions, if you just take out the neurons, you don't kill the axons going through it, cause coma. So the next question we had was, what activates the basal forebrain? What keeps the basal forebrain awake if in normal animals? And so we decided to trace the inputs to the basal forebrain, to the medial septum diagonal band and the substantia nominata. And what we found is a population of previously unappreciated neurons. This is at the level of the parabrachial nucleus. This is a cell group that surrounds the superior cerebellar peduncle as it enters the brainstem just before the pedunculopontine tegmental group. So it's actually just caudal to the pedunculopontine tegmental nucleus. And it turns out these neurons have the mRNA for the vesicular glutamate 2 transporter. Now, vesicular glutamate 2 transporter is the transporter used to load glutamate into synaptic vesicles. So these are glutamatergic neurons. So these glutamatergic neurons in the dorsolateral pons, not part of the locus coeruleus, not part of any of the monoimmune systems, not even on the map for the arousal system, have this major input to the basal forebrain that people had never really explored. And we wanted to see what would happen if we removed those inputs. So Patrick Fuller, in the lab, when he was a postdoctoral fellow, did this experiment where he placed lesions with ibotenic acid, which is a non-selective cell toxin, and he killed small parts of the parabrachial nucleus, or he placed large lesions that killed all of it. The small lesions caused about 20 to 30% less wakefulness. In the animals with the large lesions, the cortex showed no FOS at all. The arousal systems still were active, but the animals were in a complete coma with an EEG, again, in the 1 to 3 hertz range. And a continual monotonous coma that lasted as long as we kept them alive. We keep these animals alive about a week by giving them intraperitoneal injections of glucose and saline, and they never woke up again. So that suggests that these parabrachial neurons are really pretty important for maintaining a waking state. But we were worried in these animals because they weren't eating, they were unconscious for such a long time. At first you put them under anesthesia, then they never wake up and they never eat. We're worried about their metabolic status, and we actually looked at their blood chemistry. But having read chapter 5 in Stupor and Coma about metabolic coma, we were concerned about the possibility that there was something we weren't measuring, some hepatic or renal toxin that was making these animals comatose. So we did it most recently. Satvinder Kaur, who's currently an instructor in our department, did this experiment shown here. I'll take you through it because it's kind of an interesting approach to doing neuroscience. It's, I think, the next wave of things that people are going to be doing, and it may actually have applications for humans. So she used something called the ivermectin channel. And ivermectin, most of us as clinicians are probably digging through our memory. I've heard of that somewhere. Didn't they use that for worms or something? And the answer is yes. It's used to kill worms in both people and animals. It's a drug that was serendipitously found to be an anti-worm agent. And around the late 1990s, people tried to figure out how it worked. And they found that actually it's a ligand for a receptor that's found in worms, but not people. And this receptor is a chloride channel, and it has two subunits, an alpha and a beta subunit. And when you give a person ivermectin who has a worm infestation, it opens up chloride channels that hyperpolarize all the neurons in those animals, and they become paralyzed, and they can't eat, and they die. And that's how the drug actually works. Now, those receptor channels, receptor subunits have been cloned, the alpha and the beta subunit, and they've been placed into adeno-associated viral vectors. What are those? Well, AAVs are viruses that are neurotropic, but which normally are not pathogenic for humans. Usually the AAVs do not cause any infection at all unless there's an ongoing adenovirus infection at the same time. But as it turns out, you can use the shell of an AAV, just the coat of the virus that's shown here in blue, and you can sneak into it whatever genes you want inside. And it acts like a stealth vector. When you inject this into the brain, it injects its gene into neurons and disappears. It completely disappears. And when you do that, we've done the experiments to show this, there's no inflammation, there's no infection. It's like a complete stealth bomber. It comes in and does its job and leaves, and it's gone. And what you get are neurons that are expressing a gene that they had never seen before. And this will work in any species, including humans. There's been several trials using AAVs to deliver genes in humans. And so we put the two subunits of ivermectin channels into an AAV and injected it into the parabrachial nucleus bilaterally. Now, what that does is nothing to the animal at baseline. This animal is a completely normal animal. It does nothing different from any other animal. Normal EEG, normal wake-sleep cycles. However, if you give the animal ivermectin, it opens these chloride channels, and the neurons become hyperpolarized. So this is what happens if you hyperpolarize the neurons in the parabrachial nucleus acutely. So in this experiment, this is an animal, a pre-injection. This is just a normal day. This is the dark cycles. This is the wake period of a rat. This is the light cycles. This is the sleep period. And up the very top is the hypnogram. Can't get this thing. There we go. So this is called the hypnogram. The top line represents wakefulness. The second line represents non-REM sleep. So the animal's mostly awake during the night, mostly in non-REM sleep during the day. And the third line represents REM sleep. Very little during the active period, a lot of it during the sleep period. The second line here represents the delta power in the EEG. So this is the amount of slow waves in the EEG. And you can see that there's very little of it during the wake cycle. It occurs during the periods when the scoring shows sleep. That would make good sense. And then during the sleep period, which is the light part of the day, let's see if I can get this to work. It's not working. Let me see if I can use the pointer to show this. So during the sleep period, that's the light part of the day, you see a lot of delta power in the EEG. The red bars are REM periods, when there's very little delta power. And that's a normal rat's day. I don't think we need to look at the next two lines. Down here is the same animal after you give it ivermectin at the start of this recording period. And the animal is in slow wave most of the time when it should be in wake during that part of the day. That has an enhanced amount of delta activity, and that amount of delta is high throughout the entire 24-hour period. Now, it turns out ivermectin is a very long half-life. Its half-life is probably about 22 hours. So it's not a big surprise that this animal takes about 3 days to wake up, which is what happened. And during this period of time when it's asleep, it has high levels of delta activity and is not wakeful behaviorally almost all of the time. It barely wakes up a little bit, moves a little bit, maybe drinks some water or eats, and then goes right back to sleep. And it does that for 3 days after the ivermectin. So it isn't due to, and if you give ivermectin, we don't show this here, to an animal that doesn't have this AAV injected, nothing happens. The animals are perfectly normal wake-sleep cycles. So if you inhibit the parabrachial neurons acutely, animals can't maintain a wakeful state. So there's two new pieces of circuitry here. The parabrachial nucleus and the basal forebrain that weren't necessarily thought to play important roles in this arousal system, but which our current studies suggest actually may be the two most important parts of the arousal system, since they're the two parts you can't do without. If you lose either of these sites, you end up in a deep coma irretrievably. Interestingly, the parabrachial nucleus also gets a major input back from the ventrolateral preoptic nucleus. I won't go into the details of that with you. So the bottom line is that we have additional circuitry we've added here. In addition to the classic arousal nuclei, we've added this parabrachial system. And the parabrachial system projects to the basal forebrain and medial septum, which in turn project to the hippocampus and cortex, which in turn are responsible for behavioral wakefulness as well as EEG wakefulness. And this seems to be the major system that keeps this going. So life comes full circle, and when Fred Plum was near the end of his life, as many of you may realize, he had a primary progressive aphasia, and he was unable to complete the 4th edition of this book. And so he and Jerry Posner invited Nico and I to co-author with him, and we were able to make the 4th edition a reality, which was an honor for us to work with him and with Jerry Posner. So I will end with the acknowledgments to the two mentors that I had who started me on this path, and to the collaborators along the way on these studies, including the last line folks who are still in the lab, and to the institutes at NIH and the Mathers Foundation, which sponsored the work. Thank you. In the interest of time, let me ask Nico to give his talk, and then at the end we can answer questions together. So I've known Nico about 12 years, something like that. I met him, I think, shortly after or during his residency at Cornell, and Fred Plum introduced me to him as his star. And that was definitely true. Nico trained at Cornell in medicine and in medical school and did his residency there. And he had previously done his MD PhD work at, I'm sorry?
No PhD.
I'm sorry, I thought you did a PhD as well.
Postdoctoral.
It was a postdoctoral period. Is that when you worked with Purpura?
Yeah, that's, and Victor.
Right.
All my basic training is postdoctoral.
I apologize. I thought it was a PhD. Well, it should have been. They owe you one. And he got interested with Dominic Purpura in the thalamus and the role of the thalamus in behavior, and continues to work on that to this day. And thankfully doesn't beat me up when I beat up the thalamus. But Nico has been a terrific colleague and collaborator on the book, and he will tell us about his work. He's very well known for this work now, and he's actually given several talks at Harvard Medical School in various grand rounds on this. But for those of you who haven't had the opportunity to hear them, I think it bears repeating. This is really beautiful work on the remnants of contents of consciousness in people who've had brain injuries. Nico.
Hi, it's a pleasure to be here. Thank you guys for coming out so late to think about consciousness. Now, my talk is going to focus primarily on Chapter 9 of Stupor and Coma. You've heard a lot about Chapter 1 and a little bit about Chapter 5. And Chapter 9 of Stupor and Coma is about disorders of consciousness, prognosis, and patterns of recovery. And Chapter 9 is already out of date, I'm sad to say, but it's a sign that the field has moved quite a bit in the last 5 years since the book was written. And what I'm going to today, I'm going to focus my remarks primarily on some clinical cases that I thought would be interesting for a mixed group of psychiatrists and neurologists to consider. And for those of you who did hear the talks earlier last month in Boston on deep brain stimulation in our work that includes animal work in the thalamus, I'm not going to talk about that tonight, which hopefully is okay, and I think you've gotten a very good understanding of the basic anatomy and physiology of wakefulness from the prior talk. So what I want to tell you about is our evolving understanding of the problem of recovery of consciousness after severe brain injury. And what's emerging is a better understanding of something I think most clinicians who've been engaged in looking at patients who've had severe brain injury have understood for a long time. And that is that the process of recovery can occur over a surprisingly long time period. It can supervene over a wide range of etiologies of brain injury. And it's something that's hard to predict and we need to understand better because it's obviously consequential to be able to predict when good outcomes are or are not excluded, when patients who appear to be unconscious are or are not unconscious. And you know, it's harder than we'd like it to be. And I think my talk today is probably going to, you know, make that more obvious maybe than it even was before, because my impression so far, and I'm sure that we're going to get better at this, is that the more we look, the more we're learning about what we don't really know. And so I'm going to focus primarily on some use of neuroimaging tools for the assessment of level of consciousness, and focus primarily today on evidence of preserved higher integrative function in some patients that can be identified, but also the challenges of this approach and its limitations. I think it's really important because there's a lot of hype in the media that you'll hear about this work to recognize that this is really still research. None of this stuff is truly ready for clinical practice, and I think you're going to see, I hope, why I'm saying that as a result of just you know, going through this exercise. I'll hopefully have time, and I don't see a clock in the room, so I should probably have something to guide me. What time is it and how much time do I have? Because okay, so I'm okay? Yeah, okay. Because I would also like to show you a little bit about how electrophysiologic techniques can be used to complement neuroimaging technologies and some of the work we're doing in that direction, and then have a few conclusions. So just to get started, if one does look into Chapter 9 of Stupor and Coma, you'll start to see sort of a layout of the nosology of disorders of consciousness as they arise following coma. And this diagram is just to sort of orient us. Cognitive function ranges from total functional loss of cognition on the left horizontal axis and normal cognitive function at the right end of the horizontal axis. And the point of the slide is to make the observation that cognitive function and motor function are often dissociated following severe brain injury. So if normal motor function is here in the upper right and total functional loss of motor output is down here, we see that across the range of coma to locked-in state, which is not a disorder of consciousness, and there's an asterisk there to make that, remember to make that point, patients may not look very different. They may only look different, and they may only become identified to us if they show some evidence of consistent goal-directed movements. So patients that we identify as in the locked-in state are always up in this little box here. And the territory here isn't to indicate a demographic distribution, but just to sort of lay the terms out. Coming back over to the left, vegetative state is really no different than coma, except for the periods of opening of eyes and closing of eyes that are never associated with electrographic sleep-wake phenomena. Typically, and the literature is only emerging on this, but I think it's certainly our experience, and it is in the published literature, the recovery of elements of sleep-wake cycles usually only appears somewhere within the next range of recovery, which is the minimally conscious state. Patients in the minimally conscious state on their lower border show some evidence of awareness or response to their environment. They may track, they may fixate, or at a little higher level, they may start to follow some commands. The upper boundary of the minimally conscious state, which is this other bright line, the green line here, is functional communication. So patients who can reliably communicate, and operationally this is usually done by having consistent, reliable responses either through spoken language or gestural communication to indicate accurate exchange of information to questions like, am I touching my nose? Am I pointing to the ceiling? That sort of thing. And from that point on, there's a range of cognitive impairment, something called the confusional state, which I don't have on the slide, up to some point in which patients can be identified to have normal psychometric function on standard psychological batteries. But the point about this layout is to also point out that if in fact patients don't have consistent goal-directed functional movements, at this point the gray zone or the ambiguity of where they're at, depending on what information we have, if we have information that they can follow commands, then we typically think of them as just in the minimally conscious state. But we don't actually know if unreliable command following is a motor problem or a cognitive problem in any individual case. And unfortunately, we also can surmise that if we have no goal-directed functional movements, we might have patients who harbor fundamentally normal cognitive function. So this is kind of the range of problems that one can deal with, and also there are examples, and I think that they're no different than this example where patients show absolutely nothing at the bedside, and yet they may fall into this box as well, and they of course would become misidentified as vegetative subjects. But I think it's important to point out that most patients in this condition of MCS, about 40% of them get identified as vegetative, even if you are able to identify something if you're a skilled examiner at the bedside. So the problem is fairly generic. So I'm going to focus primarily today on this question of what can we do to sort of get out of this gray box. The other question that is very important in this area, I want to tell you about a little bit before I start, because I think it's just a good update your medicine, and it also is an update to Chapter 9 because it's not in there. And that is that our understanding of how people move from conditions like minimally conscious state to greater recovery has really continued to change over the last several years. And just to briefly kind of give you some of this information, because I think it is useful, and there are many clinicians in the audience who will encounter patients in minimally conscious state or vegetative state. This study, which was published in 1997, was the first of two prospective cohort studies, which up until recently were the best data we had. They involved 50 to 30 patients in the studies, and they looked at the evolution of outcomes for patients who started in vegetative state or minimally conscious state at 1 month after an injury, and then looked at their outcomes at 1 year. And in this particular study, vegetative state and minimally conscious state were divided into etiologies of traumatic brain injury versus non-traumatic brain injuries. And what Giacino and Calamar found in this study was that, in general, patients with traumatic brain injuries, whether they were in vegetative state initially or minimally conscious state, showed better recoveries. But patients who began in minimally conscious state of either etiology, but particularly for traumatic brain injury, showed significantly better outcomes at a year. And in fact, patients who remained in minimally conscious state after traumatic brain injury at a year or 3 months included outcomes of no disability at 1 year. So this is a good outcome. This is the kind of thing you don't want to exclude when you're prognosing or talking to a family in the hospital. And I think probably most people start to get a little nervous when they realize, gee, you mean 40% of people who are minimally conscious could be misidentified as vegetative, and I might see somebody who's vegetative by either a report or by the exam I can do at 3 months and is still an inpatient. and they could still have a good outcome at a year. I'd kind of like to know about that. And actually, it turns out this is the norm. So 80% of people who end up getting into rehab, which in minimally conscious state, which is about 4 months entry into most programs, 80% of them will emerge. Now, not all of them will end up with no disability at a year, but many of them will end up with severe to moderate disability and have elements of independent daily living activities and more. And so now, and this I think is really, it's like an ethical obligation that I share this with you guys. This study just came out. It's the largest cohort that's been done. It's only traumatic brain injury patients, but it looked at 400 patients that were prospectively enrolled in the Traumatic Brain Injury Model Systems Program, which is run by the National Institute of Disability Research. And this group, if you look at these statistics, is really kind of impressive. These are 400 patients, median age of 28, all Glasgow Coma 3 at onset of their coma. These are very severe injuries. These are not Glasgow Coma 8 patients. They're evaluated at 1, 2, and 5-year time points. Like all studies before them, their entry into inpatient rehab is somewhere between 2 and 4 months. And these are their outcome statistics. 68% of them recover consciousness. 23% emerge from post-traumatic amnesia. And of that group of 91 patients, 66 regain independent living without in-house supervision, and 63% have employment potential. They regain vocational reentry. Okay, so this is like, these data actually don't comport very well with the way in which patients are given opportunities to convalesce and also have access to some of these kinds of programs. There aren't too many of these programs. I think there's 7, maybe 9 in the country right now. One of them is here at Harvard. Joe Giacino has just started it at the Spalding Center. And then the final piece of sort of update of Chapter 9 is that there is now the first evidence of any agent, first class 1 evidence for any agent to improve outcomes in the subacute or chronic phase of recovery following vegetative state or minimally conscious state, and that's, again, only after traumatic brain injury, and this is in the New England Journal, published earlier this year, showing that amantadine hydrochloride has an improvement effect when given during rehab, and it's enough of an effect to create a significant difference despite the fast rate of recovery over the first year that I showed you is present in this population of patients. And most people have probably had this topic introduced to them, as I did in some ways, by cases of very surprising late recovery, and they of course are also true and part of the story. Not too many of them, but this man here, Terry Wallace, is the longest on record that I'm aware of, and we had a chance to study him several years ago. He spontaneously emerged from minimally conscious state at about 20 years after his original injury. So when we talk about a slow process and one that doesn't have a lot of really firm understanding, these are sort of the boundary conditions, the metrics, and then sort of what's common to the cohort. So that's information I think everybody should be aware of. So how do we start to do a little better? One way we can do better is to make measurements of brain function early on to try to sort the variance here and understand kind of where we might be on that 2D diagram. And more or less, this kind of work has been divided into two areas. One is neuroimaging techniques, and the other is electrophysiologic techniques. So let me just focus a little bit on the neuroimaging techniques to begin with. Probably the iconic image that most people are aware of is this study from Adrian Owen and his colleagues published in Science in 2006 of a patient who clinically was identified as somewhere between minimally conscious and vegetative. They showed some fixation at the bedside when these images that I'll explain in a moment were taken, and then following a period of about 4 or 5 months, they were studied first 6 months after their traumatic brain injury. They then made clear transitions into the minimally conscious state. However, those bedside exams were very dissociated from what the neuroimaging work in this subject showed, which was that if they were asked to do mental imagery tasks, either imagine themselves playing tennis or walking around in a familiar environment, they showed activation patterns in expected regions within the brain using the functional magnetic resonance imaging technology that normal subjects would show. So here's tennis imagery. Imagine yourself playing tennis. An area in the supplementary motor area of the frontal lobe activates in control subjects, and here's in the patient. Or walk around your house, and this is a normal subject control group result, and this is an individual result from their patient. So this is a demonstration of the problem of identifying somebody who looks at the bedside as if they're here, but their range of cognitive function is really undisclosed and undetermined by the measurement technique. This technique also invites the idea that one might be able to convert this sort of measurement into a communication channel. And in 2010, Martin Monti and the groups in Liège and in Cambridge published a paper in which a single subject was studied and evaluated using these technologies and demonstrated the ability to answer some yes or no questions, 5 out of 6, about personal history accurately by carrying the instruction for yes to play tennis, for no to imagine walking around your house, and then go through a sequence of these kinds of steps of imagining the activation to act as a proxy for a yes-no response, either spoken or generated through a gestural command. Now, that sounds like it would be great, and this is just going to work. But and it will in some patients, and one can always have a proof of concept to demonstrate this. And if the patient was locked in and not structurally brain injured with what looks like a hydrocephalic brain, we wouldn't even be surprised to see these results. But we have to calibrate kind of our thinking about who are these patients, what's going on with them from a pathophysiologic point of view, and how do we start to look at this entire area and match an approach that can allow us to see where these techniques come in and what some of their limitations are going to be. So we set out to do that. John Barden, who just completed his PhD in my lab, did a very nice thesis beginning with neuroimaging technologies and fMRI, and then moving on to some quantitative EEG work. And he began with a variation on the tennis theme of imagining yourself swimming. And as you'll see in a moment, the reason we did this is that we had an index patient who we were interested in studying who was a swimmer, not a tennis player. And as was known from the literature before the Owen study was carried out, and there's a long literature on this in imaging, it doesn't matter that much whether you're playing tennis or swimming. The supplementary motor area is typically the area that activates for most of these motor imagery tasks. This is our normal group. This is one of our normal control subjects. We were able to show that more or less that is what we should see. And we began to apply this approach for both command following communication to patient subjects. And now, admittedly, I knew there were psychiatrists in the audience, so I felt like I should do something like this and it'd be late in the night and keep everybody on their toes. And so I'm going to give you a clinical intuitions Rorschach test. And I think probably, I'm not sure how much more of the talk I'll get through besides this, because I don't know, how much time do I have? You said 8, but no, okay, all right, thank you. So here's the test. I'm going to show you 2 patients, and I'm going to give you most of their clinical history, but maybe not all of it till the very end. And I want you to start to form your intuitions as a clinician about what you're seeing. Okay, so the first patient I'm going to present to you is a 22-year-old female. She had an idiopathic occlusion of the basilar artery. You see here is the stent that was placed acutely at the time that her artery occluded. She had an extensive basilar pontine injury. This is the T2 image, this is the T1 image. These are relatively close to the midline parasagittally. As you move more laterally, you can see that there's continuity of the brainstem. The injury extended into the thalami bilaterally into the central regions of the interlaminar system, and also on the right side involved the lateral geniculate body. The right occipital pole was involved, and there was some evidence of involvement of the entorhinal cortex on both sides as well. So a very extensive occlusion, top of the basilar component, and basis pontine injury from a basilar artery occlusion. Her diagnosis was vegetative state or low minimally conscious state for 2 years. A very astute neurologist in, in the audience, I don't know if I should identify him or not, Dr. Katz, had noticed that the patient could track vertically with the left eye. And this unambiguously is a sign of minimally conscious state, not vegetative state. Although, interestingly, both the family and the facility that was caring for the patient at the time we first saw them had the patient coded as vegetative. And the patient's family's number one question to us when they came for our first evaluation of this subject 5 years ago was, is there any evidence of awareness in this person? Because from our point of view, even though we've been told that there's something related to minimally conscious state, we don't see anything different than what we would think vegetative state would mean. The patient on our exam 5 years ago showed periods of spontaneous eye opening of the left eye, and we established intermittent control of downward eye movement. And this was the first demonstration of an eye movement to command at that point. So this is the first patient. Now, second patient. Second patient, that is patient A, this is patient B. Second patient is a 27-year-old male who sustained a severe diffuse axonal injury with predominant axial involvement of the brainstem. He had a T-bone motor vehicle accident, so his car was crossing an intersection, was hit, and then spun. And he had lots and lots of radial injury. And you'll see this better in one of the next images I show you. But you can see he has ventricular dilatation. In this image, which is sort of out of plane, you can't quite appreciate how much brainstem injury there is, but you'll see it in a moment. But his injury was global. His diagnosis was vegetative state for 3 months, and the family reported evidence of visual fixation at 6 months. We examined him 3.5 years roughly after his injury, and we noted head movement to command. Now, these two patients were brought into this study of command following and communication using imaging. And we did fMRI and we did EEG. So before I start to show you the data, I'd just be curious, by a show of hands, how many people think we can establish evidence of command following or communication or in either of these patients using the fMRI technique? Okay, good. So everybody's sort of with me. It's probably worth trying. If we can demonstrate that kind of finding in fMRI, how many people think a systematic communication system could be developed for the patient? Just in general, of any kind. Okay, so most people, right? And how many people feel their answer is, better than the impression of an ink blot?
I thought that was the fMRI.
Good. Okay, so let's look at the data. All right, so this is our subject A with the brainstem injury. And this is her best response. I should say that this is the response acquired on the second of 2 consecutive days that we tried to image this patient. We brought the patient in for several days to observe them and to do these studies. When they first showed up, their percutaneous gastrostomy tube was clogged. They couldn't receive their dose of amantadine, and we did not get a reliable response in the fMRI. However, the next day, when they had their standard medication dose after the tube had been reopened, recannulated, we got a very robust, very clear response. And it's in the supplementary motor area, but it's also in other areas of the brain. And for those of you who are familiar with sort of bold responses, if we look at the voxel here in the parietal lobe, this kind of response, the blue is imagine yourself swimming, stop, imagine yourself swimming, stop, imagine yourself swimming, stop, continuing on. This is probably better than most of the normal subject data that we have. This is very, very, very robust and very clear. This is the other patient. This is their command following data. Now you can appreciate a little bit better the atrophy of the ventricular system, the brainstem damage to this patient. The signal is noisier. It nonetheless passes the statistical criterion and is in the right area. There's an SMA response. So both these patients had fMRI command following signals. However, when we went on to the next step to try to establish communication, and in this study we did 2 things. We did yes-no communication, just with the single response, not with 2 different responses, and multiple choice communication. I'll show you this in a minute. This patient, despite the fact that they could do this, was unable to generate a response in any of the communication paradigms. Okay. The other patient, I'll show you in a minute. So this is the multiple choice version of the task. And in the multiple choice version of the task, we made this very, very difficult. In this case, we showed individual normal subjects a playing card. We asked them to remember their card. A Confederate uninvolved in the analysis presented the card to them, so only the Confederate and the subject knew the card. That was true for the normal subjects and the patient subject. And this, like everything else in fMRI with normal subjects, was trivial. You did this in 5 minutes, you went into the scanner, you did this, it was easy to get the result off the console, and we just do this. That's no patient does that, except for one patient, and that's the first patient that ended up in the literature in the New England Journal paper. And this is the fine print that's not in the paper, but it's very important. The reason that the 5 patients who did command following, only one patient in the New England Journal was studied for communication is because Audrey van Hooidenhuizen in Liège, Belgium, who was studying that patient, saw at the console that the patient was doing the command following. And the quote was, "Oh my God, this guy's doing this." And then they went on and did the sequence of questioning. And this is a historical fact about this, because in fact, we gave up on doing real time at one point because it just became impossible to convince ourselves that we could separate signal and noise, and we didn't want to sort of spend time trying to do that. It is possible to do that if you have strong responses, but not every patient has very strong responses. So here are the answers to the normal subject, and this was their card, the Ace of Diamonds. But now this is what we got from the patient with the brainstem injury. Her card was the Ace of Spades. At the bedside, when we presented her with her card, she could look down for her card and did not look down for distractors, but she also had periods of time when she didn't control her eye and didn't respond at all. For the face card, imagine yourself swimming if your card is an ace, imagine yourself swimming if your card is a jack. She showed a reliable response only to the jack, but the card was an ace. For this, for the suit of the card, we saw something for club and we saw something for spade that passed the statistical criterion using this approach. And we wondered if, in fact, what might be going on was that the patient was having a delayed response. So if we signal average the hemodynamic response for our normal subjects, we saw a very different shape to the bold signal response than for the patient subject. And this was also reported for the patients in the New England Journal article by Monty et al., that there was a hint that there was a bimodality to the response and that it might have been a little slower. Our response window, because we had the 4 multiple choice conditions, was also shorter than 30 seconds. It was out to 21 seconds. So it was possible that we were looking at a delay. But it was hard to connect that with the data I just showed you, where there were 2 responses on one run and 1 response on another. So John Barden, in part of his thesis, got very interested in work that was being done in fMRI with something called multivariate pattern analysis, MVPA. And he tried to use the multivariate pattern classification technique on the whole MRI data cube. And first he tested it in normal subjects and found that if he looked at the whole brain response using one of these MVPA analyses, he actually did do better than the region of interest method for the normal subjects. And when he applied this to the population of patients that he had studied, he found that the same thing was true for the patients who had shown a response with the standard Roy-based methods. And so what he then did was to use the pattern classification algorithm to take the pattern from the patient's command following response when they were just doing motor imagery and use that to test the communication runs. These are 3 different experiments. So to understand what this patient had to do, they had to stay in the scanner for about 45 minutes and carry out 3 separate experiments to do these things. It's actually a lot of effort, right? And what he found was that if he looked at the question about face, the JAK clearly was the only response, and both methods, the GLM method and the MVPA method, concurred. But when he applied the MVPA method to the suit, unambiguously, only the club showed that it was a result that was reliable. And so now what we had was 2 independent experiments that concurred with the hypothesis that there was a delay and that the patient was trying to systematically give us the right answer. Of course, one has to draw an inference like that. You can't actually be sure. So now I've given you some data about all these patients, not all the data. And I want you to think about this question, and then we'll vote. So choose by a show of hands the clinical outcome in these 2 subjects over time. So let me just read the choices, and then we can vote on which one you want. Outcome 1, neither patient was able to establish a reliable communication system using some sort of augmentative communication device over time. Outcome 2, patient A, the one with the brainstem stroke, but not patient B, the one with the diffuse axonal injury, established reliable communication using augmentative communication systems. Outcome 3, patient B, the one with diffuse axonal injury, but not patient A, the one with the brainstem stroke, established reliable communication using augmentative communication systems. Outcome 4, both patients established reliable communication using augmentative communication systems. I think this question's a little harder. So how many people want to vote for 1? Neither patient was able to establish a communication system. A few votes. How about, who wants to vote for number 2? The brainstem, a lot of votes. Who wants to vote for number 3? A couple votes. And who wants to vote for number 4? Okay, so this is a non-scientific poll, but I will tell you that the majority of you voted for number 2. The next largest number probably voted for number 4. Somewhere between 1 and 4 were about equal, and the smallest number of people voted for number 3. The answer is number 3. Okay? That's why this is going to be hard. All right? That's why this is going to be hard. Number 3, what I didn't tell you was that number 3 came to us at 3 years, and although for a year only the family and 1 caregiver had identified anything above the vegetative state, the 1 caregiver was right and could see that the person was answering questions reliably and got to the point where they could use a letterboard with their head movements. And when the family brought the patient to us, they were trying to get a BCI from Medicaid. Medicaid had provided them with a 12-week trial of a system that would have a head mouse, but the system came to their house with a broken USB port, and Medicaid would not pay for the person to come out to fix it. So our enterprising medical student, who's now a Johns Hopkins neurology resident, found a head mouse on the web, a webcam head mouse, gave it to the patient, and the patient went home with it, and it started to work. And about a year later, we got an email saying, not only is this really working, but this email that you're about to get is from the patient. And he had been able to start using his head to use a communication device. Then sometime about a year and a half after that, for reasons I think are interesting and maybe related, suddenly the patient started to have enough movement of his hand to be able to use a joystick. So now that patient has independent movement of hand, writes emails, runs around in a little wheelchair and sends us communications, and has initiation and reliable communication through the augmentative communication systems. Unfortunately, we are still struggling with the patient with the brainstem stroke, and Doug and others we're all looking for a reliable method to link into to this patient, and we think we may be on the trail of something, but this is now 5 years. So it's not going to be easy, and I think this is something that people have to realize, that a single patient who does this, like a locked-in patient, we shouldn't be surprised when we see that. It's not actually going to solve the problem of understanding how to find these patients who have a very, I think, high urgency to treat and figure out what to do for them when we do find them. But one last thing, and I'll do this quickly, is that fMRI is hard. fMRI, putting patients in fMRI machines and getting reliable signals because of motion artifact, lying them flat for long periods of time, the noise components, not a very easy technique to use. So everyone wants to see electrophysiologic methods come to bear to sort of do this at the bedside and hopefully at least be able to sample it more consistently. And, can I do 5 minutes on this, or should we? Yeah, okay, I'm just conscious of that. I'm going to give you guys a very brief tour of quantitative EEG. This is a slide courtesy of my colleague John Victor. It's a very nice way to do it. These are the traces from Penfield and Jasper's monograph in 1954, which was the first book to kind of relate EEG patterns to states of consciousness. What they called excited was really the desynchronized EEG in wakefulness. Relaxed was really sort of an eyes closed alpha. Drowsy was a theta rhythm, so sort of not a great drowsy trace, but that's what it's supposed to represent. And then different stages of sleep. This is light sleep with spindles and a K complex. Deep sleep here is kind of an awful looking tracing, and coma is just a slower tracing. So when the quantitative EEG person talks about the spectrum of an EEG, what they're really trying to summarize for you is the frequency content of the dominant frequency content of a signal like this. So some signals have a mix of frequencies, but some signals are really dominated by one frequency. So we know if this is the alpha rhythm, it's going to be something between 8 and 12 hertz. This is about 10 hertz, and a frequency spectrum would show you that most of the signal power is concentrated around 10 hertz. For a drowsy EEG, something like a 7 hertz rhythm may dominate it, and we'd have maybe a broader peak because the frequency is not so isolated. For the deep sleep tracing here, maybe 3 hertz, and then for these very slow waves with a lot of DC power for the representation of the comatose patient, maybe something that looks very much like the tracings Cliff showed you of the comatose-like state in the rodent after the basal forebrain injury. For the states that have a mix of frequencies, in the wakeful state, there's a 10 hertz rhythm, which is not quite as prominent, and then a broader range of higher frequencies in the beta and gamma range, and the spectrum might look something like this. For the sleep states, which have spindles, which are a 15 hertz fast rhythm, and then slower components, it might look something like this. So what we've done to look at the EEG equivalent of the command following communication paradigms I just showed you is to just simply transfer them wholesale into the EEG to start. And this is work by Andy Goldfein in my lab. And Andy basically took the exact same paradigm that was used, timing and everything, from the MRI experiment, and collected data in normal subjects and patients from the same types of tasks. Imagine yourself swimming, stop imagining swimming. EEG following the command was taken out, looked for artifact-free segments, and then spectral analysis on groups of clean segments concatenated from multiple runs, typically 8 runs of imagine yourself swimming, stop, and then as many clean seconds as could be cut out of the middle of each of those runs were concatenated into large data arrays and then analyzed. And here's the kind of result that you would get from a normal subject. So these are a couple EEG channels. This is the central channel in the back of the brain, OZ. This is a channel over the left central parietal region, CP5. And these are spectra. And what you see in these normal subjects is there's a resting alpha rhythm in this subject that modulates maybe a little bit, but the main signal is a suppression of power here between about 12 and 16 hertz. In this subject, there's actually an elevation of power in the back of the brain in the alpha range when the subject begins to imagine swimming. And then he's displayed where there's a statistically significant range of frequencies separating on these complex arrays that show all the electrode channels over the head, and where there's significant differences in the spectra from 4 hertz to 24 hertz. And what you see is there's sort of a nice spatial pattern. It's robust. It has consistency across more than one channel, and there are an array of interesting statistics you can apply, but these are not close calls. These are very clear and robust responses. What Andy then went on to do was to look at all of the patients in whom we'd identified command following responses with fMRI. And this is the patient that we just looked at, the one who communicates reliably with the diffuse axonal injury. And this patient had a very robust response. Interestingly, this patient's response is the reverse of all of our normal subjects in that they synchronize in the 12 to 16 hertz range instead of desynchronize. In other words, there's more power with swimming in this range and most of these channels that showed a difference than there's a suppression. But that's actually a normal variant, it turns out. It's not necessarily related to any element of plasticity. This is something that recent work shows is in the normal population as a tendency for some people to synchronize versus desynchronize with this type of activity. But in this subject, the EEG and the fMRI were very reliable for the command following. The fMRI was not reliable for communication, but the patient's bedside communication was quite robust. I don't have a slide for any of the other patients because we don't have robust, reliable EEG from them, even though they had identifiable fMRI responses. This is another thing. These are independent measurements. They're going to have their own sources of variance. They have to be done very carefully. We have some hints in one of the patients, but I'm not going to show you the hints because I think what you really need for this is for it to be gangbusters clear. Otherwise, it's not actually going to have that much value except for maybe some diagnostic hints. And finally, it may be possible to do simpler things that will help us identify when patients are changing and as they're starting to evolve lower levels of recovery from vegetative to minimally conscious state that don't necessarily entail this very high level cognitive function. And just to give you an idea of what this looks like, this is another study from John Barden's thesis looking just at spoken language presented as unintelligible speech versus non-intelligible reversed language and baseline EEG. And in normal subjects, spoken language tends to desynchronize alpha rhythms. In this one clinical subject, John found he was actually able to demonstrate that spoken language in this particular subject not only was different than reversed and baseline, but shifted the entire spectrum, at least in this one channel, from a low frequency theta rhythm to a 10 hertz alpha rhythm and some higher frequency components. And in this case, and this was something, this was sort of the general phenomena of desynchronization with language stimuli was something that John found distinguished, at least in a very small subset of patients studied, vegetative patients from minimally conscious patients. But very interestingly, in this one subject that I just showed you, he was able to show that in real time, one could actually observe this process. So here's a trace of 1 hour from this patient at the bedside. The nurses talking to the patient suppress this low frequency oscillation that goes away with other language stimuli, such as the investigator speaking to the patient or task instructions, but doesn't modulate to the family entering the room or machines beeping. And when they hear spoken language forward, you see it goes away completely. But when the spoken language is presented to them in time reverse, it doesn't modulate. So, these are the kinds of tools that could be further developed and used in conjunction with clinical exams, hopefully, to improve our diagnostic acumen as we go forward. And I'm just going to, say a few things in conclusion. I think the implication of what I showed you today is that longitudinal assessments of severely brain injured patients really are required periodically. Right now, only the Defense Department is suggesting that patients get yearly examinations to have their diagnosis reevaluated. This was something the IOM put together an exploratory meeting to discuss, among other aspects of these disorders of consciousness questions, and none of this has really kind of gone forward with new policy recommendations. They couldn't even get the study funded that they recommended for further exploration in this area. I think it's clear that there are clinical contexts in which there's going to be a higher risk of having higher level function that's masked when you have brainstem stroke, possibly with striatal dysfunction following injuries that impair the outflow from the medium spiny components, or patients like the patient I showed you who have a prominent axial brainstem involvement and diffuse axonal injury. And in general, the problem's going to get worse because everything we're doing is improving the probability that more neurons survive rather than die after acute injuries. And as this trend in ICUs continues, I think we just have to get ahead of stratifying our patient population a little bit more precisely. And then finally, you know, we're seeing really, I think, new syndromes. I mean, when you see somebody who looks vegetative and can follow commands with fMRI, it has nothing to do with the vegetative state. It's a different syndrome, probably doesn't have a name right now. It's not locked in state unless they more or less can do everything as if they were fully conscious, and that hasn't been demonstrated in the literature, although you know, people have identified patients who it seems like they more or less fit those criteria. And I think the highest risk medical error to be avoided and now anticipated is the possibility of a conscious autonomous agent who is unable, he or she, to exert their own opinion. And we'd like to know when we're needing to worry about that, and I think we'll do better with this as we go along now. And I'll just thank my colleagues and again, Dr. Plum, who began and ended this duet, and collaborators here at Harvard and in other places, and our funding agencies, and thank you.